Okay, hi everyone, and thanks very much. I appreciate being here, so thank you to the committee for that. My talk continues focusing on the linkages here, and it's completely focusing on the microorganisms. So we are back now to the nitrogen cycle again. This is a very nitrogen-focused session, I think, and if you think about the keynote lecture, it was a lot about denitrification, and this talk continues on that. So I'm focusing on categorizing different functional groups within denitrifiers. So denitrification is just not one function, it's a suite of functions, I would say, and I will show you that. And to address that, we have been looking at genomes. So this talk is focusing completely on comparative genomics of denitrifiers. And those are genomes that then harbor these genes that code for functions in the different parts of the denitrification pathway. And our idea is that by doing this, we might be able to interpret the environmental sequences better if we have a framework and we understand more about how the organisms are organized and how they function, and what implications that might have for greenhouse gas emissions, and in this case, the nitrous oxide. So just to recapture the nitrogen cycle, here is a very schematic picture of the nitrogen cycle, and the process here in blue is denitrification that you heard about before. So we have emissions of nitrous oxide since nitrous oxide is an intermediate in the denitrification pathway. We have nitrous oxide being produced also in other processes in the nitrogen cycle, but I will not focus on that in this talk. The interesting part is also that we have the last step in the denitrification pathway, where nitrous oxide is being reduced to nitrogen gas, and this is the only sink of nitrous oxide that is known in the biosphere. Now, we heard before that it's difficult to target denitrifiers by a taxonomic approach since they are so diverse. So therefore, we're often using these marker genes that encode for steps in the denitrification pathway. And it's very common to target the near genes that code for nitrite reduction. And there's two different types of that. So those are the common markers for denitrification. And we recently looked into 1,100 metagenomes available in databases. I think it's really slow now. And to see where do we find near genes. And in all metagenomes that's available, we see that we find them in all the different major habitats. So it's a very commonly occurring genes. And we can see that they seem to be, the nitrite reductase genes, to be very abundant in engineered systems, and this is mainly wastewater treatment plants and sediments, and it's completely dominated by sediments, wetland sediments here. When people are doing metagenomics and find near genes, it's often interpreted as denitrifiers being present in the system, but that might not always be the case, and I will come back to that at the end of the talk. But first, I want to define these I talked about different functional groups within denitrifiers. So here is the pathway again, going from nitrate via nitrite, nitric oxide, nitrous oxide, and nitrogen gas. And if we exclude the first step, because this is not at all, there's lots of other organisms that are doing nitrate reduction and focus on these key steps of denitrification. We can see that we have organisms that have the full pathway and have one type of nitrite reductase catalyzing this reaction. Then we have the other types that have the other type of enzyme for this reaction. And then we have those that do not have the last step, so they don't have the capacity for nitrous oxide reduction. And then we have the third type, then, which have only the last step, only nitrous oxide reduction capacity. So we were interested in understanding which organisms, where do we find these different functional groups of denitrification, because this will of course have implications for nitrous oxide emissions. Those, if they have the capacity, if they are also have the capacity to produce nitrous oxide, only do it or only reduce it. This will influence the net emissions from an ecosystem. So to do that, we did a comparison between 652 genomes that have some of the denitrification genes. And then we looked at if they had, which genes they had. So did they have near K, near S, the nitrite, different types of nitrite reduction enzymes, the NOR, which I will not talk about at all today, and then the pink one here, the nitrous oxide, the gene coding for nitrous oxide reduction. And then this is a tree of all the organisms we found, and based on 18 and 16S. And we can, first of all, conclude that we find these genes in all three domains. And I think the pointer is not here. We have some fungal genomes here, we have some archaeal, but the majority are bacterial. And also here, we have the proteobacteria. So the proteobacteria are dominating in the data set. So the focus was to understand more about nitrous oxide, what regulates nitrous oxide emission, the ultimate regulation, the genetic potential in the system. So first, if we look at the data set, like on a broad view, and then divide and look at the denitrifiers that have a near K type of nitrite reduction or near S type of nitrite reduction, we can see a very interesting pattern, and that is that the organisms that have near K coding for nitrite reduction very often do not have NOS-C, so more than 70% lack this last step in the process. Whereas denitrifiers that have the other type of nitrite reduction, they in most cases carry this gene also, so it co-occurs. And this is an indication that community composition would matter for N2O emissions. If we then focus on all the organisms that carry the gene coding for nitrous oxide reduction, that was almost half of the organisms in our data set. We can see that the majority of them, about 70%, that's the green here, these are genomes that we would call canonical denitrifiers, and with this I mean that they have the full denitrification pathway, so they have all the genes and then nitrous oxide reductase at the end. But interesting, we see that among these genomes, 30% actually only have NOS-C. So that's a very large fraction of the genomes that are actually potential N2O sinks. And you heard before about that this NOS-C gene that code for nitrous oxide reduction can be split into two clades. And this was presented on a poster more in detail yesterday, and the first author here, Christopher Jones, he's attending the conference, so if you're interested, you can address him for details about this. Now, if we look at these two clades, clade 1 and clade 2 of the nitrous oxide reductase, we see that we have a much higher fraction, more than 50% of the genomes belonging to clade 2 have only this last step, whereas it's a smaller fraction, only 16-17% among clade 1 that are carrying only that gene. We have a pause here. Okay, we go back to the genomes here again, and as I said before, we have these three different groups, but they are also in subgroups. So the ones that have only nitrite reduction, they can have then either the near K and near S, or perhaps both of the genes. And the ones that have a complete denitrification pathway, it's the same. They have either near K, near S, or both of these genes. And then the third group with only NOS-C. So we wanted to see how these relate, how these different co-occurrence of genes relate to the taxa in this data set. So this is a table where we had looked at the co-occurrence patterns, types in relation to taxa affiliation. And I'm showing a table where we are at the phylum level. And this is based on chi-square tests, and we are looking at all the ones that are marked with blue, means that they are overrepresented in the data set, and all the ones marked with orange means that they are underrepresented in our data set. So as an example here, we have the proteobacteria. They are overrepresented, the type having either nitrite reductase of the near K type or near S type, combined with having NOS-C. These are overrepresented, this type is overrepresented among the proteobacteria. So these are what we would call then typical denitrifiers. And we see here the ones that have only near K, so no capacity of nitrous oxide reduction, they are overrepresented among the actinobacteria, but also among the Ascomycota. So potentially these are producing a lot of nitrous oxide. And then as an example of those that have only NOS-C, we see that this type is overrepresented among Bacteroidetes, but also Euryarchaeota and Firmicutes as an example. So this means that it's not, these different co-occurrence types are not randomly distributed among the taxa that we investigated. So there is a pattern, so certain taxa where we see that we have more of one type and other taxa are more prone to belong to another type. We took the same, we did the same tests to see where these genomes have been isolated, to also have a look at if there is a relation to sort of the lifestyle or preferred habitat of these co-occurrence types. And we can see from this picture, it's basically the same table as you saw before, but instead of taxa, we have the preferred habitats here of the genomes. And what we can see then, for example, denitrifiers having near S combined with NOS-C, they are overrepresented in wastewater treatment systems, but also in freshwater systems. Whereas those, for example, having only NOS-C, they are overrepresented in marine systems among these isolated genomes. But then we know that, of course, there's a bias. These are genomes. They have been genome sequenced for some reason, and they might not be very representative of what we find in the environment. So how do we deal with the environmental sequences? Well, first of all, the information that we have from the genomes, we think that that's relevant to have in the back of your head when interpreting your data. For example, that actinobacteria do not have, and that's a rather substantial number of genomes that have been sequenced, that they don't have NOS-C, for example. But we also need to look more into the nitrite reductase genes to really be able to interpret the environmental data, environmental sequences. So we're proposing a framework for the near genes based on what we call a phyloecological approach, and I will explain what we mean with that. So we went back to the databases, and because there's so many genomes coming all the time, so we went back to the databases, collected more sequenced genomes, and gathered all the ones that had near sequences. And then we established a way to define the protein to be able to distinguish the real near genes from the ones that are not really catalyzing nitrite reduction. And this is not so easy, it's rather complicated. Then the phylogenies were reconstructed to have really robust phylogenies that we can map our environmental sequences to these phylogenies. And then characterize the lineages, and that's depending, of course, on the sequences, but also the genomic context, other traits that the organisms may have. And then we tested this framework on 1,100 metagenomes from 32 different environments. And I will only give an example of the near S type here today. And basically, the clustering we found, it has an apparent ecological clustering by clades. And we can see that we have the canonical denitrifiers in a very clear cluster. And these are the true denitrifiers, proteobacterial. We have some alternative near S, proteobacterial. And the other clades, we can see that we have denitrifiers, but others, we see the meselotrophs, for example, where it has a different role. This is not really denitrification as we know it. It's nitrite reduction, but it's not denitrification. We have in Anamox, for example, it's nitrite reduction, but it's not denitrification, and so on. And we also found some interesting stuff on the chloroflexi where it's not denitrification, but something else that I will not talk about today. So when applying this framework, as I said, to the metagenomes, it looks like this. We can see that the canonical denitrifiers, the typical ones we know, the proteobacteria, they are dominating in the metagenomes that we analyzed. We can also see that we have some metagenomes, for example, since this is a soil conference, I can point out that there are no hits for near S in forest soils, but we know they exist there by targeted approaches, but not in the metagenomes. I can also point out that we see that in agricultural soils, we have also a quite large fraction here, more than 20% of the hits that do not fit into the phylogeny as we know it. So these are unknowns that come up in the metagenomes. And we also see that in many of the different habitats here, that archaea seem to play an important role, and there's very little done on the importance of archaea in terms of denitrification. So we go back to these, the picture I showed before. We have these near genes present in different habitats, and if we would use our framework, I will just give the example of soil here. We see there's a lot of near K here, a bit less near S. And if we use our framework and look at, for example, just filter out the proteobacterial ones, it looks different. Then we don't have near K dominating, then it has switched and we have a dominance of near S instead, for example. So with that, I come to the conclusions. We have two major conclusions, that the denitrification pathway is modular, and this underpins the importance of community structure for N2O emissions. We also show that we have an ecological clustering of the near genes, and that suggests that the metagenomic data needs to be analyzed using a phyloecological approach. We can't just make inferences based on gene presence. And that might actually be true for a lot of other functional genes. With that, I would like to thank the research group, and especially Daniel Graf, Herman Bonilla Rosso, and Chris Jones that have contributed to the data that I presented today. And if you're interested in more of our work, Chris Jones is also presenting another poster today on the on ammonia oxidizers and nitrite oxidizers. I don't know the number, but you will have to walk around. Thank you very much.